with you guys getting a little messy today. We will be playing a game later. We're going to continue that game on into Wednesday, so you'll want to find each other again on Wednesday. So all in all, it's just going to be a super fun week. However, I want to start with some logistics because you do have some impending due dates. Homework number 1 is actually due tomorrow, which means that you may want to find me tonight on the conferences button. I will be there from 7:30 to 8:30. When I begin that conference, Canvas will automatically email you and tell you that I'm starting office hours. If you would like to just follow that link and find your way into my office hours, I would love to see you there. Even if you're just coming in to see how that conferences tool works and finding out if it'll work okay for you. And if you get in there and it's not working, that's a good place to start to figure out how it is that you'll be able to find me there later on. So give it a try, and I would love to see everybody there. It handles lots of students all at once, so it's totally fine if everybody wants to log in. And, of course, do so from wherever you have internet. Also, being said, that homework due date, we also have that knowledge survey and pretest going on. Some of you have already popped in there and done that pretest because you're hoping you can drop your low homework score come the end of the semester. So that is fantastic if you have done that. One note on that, it is a sort of single attempt thing. So for those of you who happen to be senior level students and you know a lot, unfortunately for you, it's going to take you longer to go through that knowledge survey because you actually know the answers, which makes it hard. Many of you will see those questions and you'll say, I don't know the answer to this, and that is totally 100% fine. Just write a note on there that says, I have no idea what you're talking about, or whatever you want to say, and then just rate your confidence accordingly. And for some of you, that might be almost every question, because maybe you just haven't had any of that material yet, and that's so fine. It is so fine. So don't feel as though that represents in any way some sort of assessment of you. It's just getting an idea as a collective, like where an incoming class is. Okay, so that's the knowledge survey. We'll look for that on the 18th if you have a chance to get that done. Now, here's what we're doing with the poster groups. So far, I've received the names of the group members and the name of your group from nearly every group, which is fantastic. I might have a new favorite group name, and it was the Probiotics. Yeah. Is that not like the best thing you've ever heard? So you guys now rank maybe above gangrene in my mind. So I'm very pleased with that. It took you a little longer to figure out your name. I think somebody emailed me like at 6 this morning, finally figuring out the probiotic name. So high five on that one. But in terms of future communications, your spokesperson has already been nominated by the group, right? That person that was the emailer to me is now your group spokesperson. I think it's an awesome idea to have group members that specialize in something particular, you know? Somebody who feels comfortable being a spokesman, a liaison to talk to me. Somebody who maybe feels comfortable leading the research aspects of things. Maybe you're just really great at dorking out in the library. You want to nominate yourself as the research leader. So I think it's fantastic if you can all have individual things that you all do well, and maybe you nominate yourself as playing a particular role in that group. I would love that. So that means the person that I communicated with this weekend, most of you yesterday, make sure that you do communicate back with your group. And one of the things that I said on that email that I sent to you is maybe you want to make yourselves a group on wild courses. You guys can actually self-construct a group there. Feel free to do so, especially if you're the kind of folks that wake up at 2 in the morning. I always do this, and I've got this great idea, right? And I sit there in bed, and I'm going, oh, man, I got to do something about this, so I got to get up and, like, write something. So if you want to have a shell, you could share with each other even at 2 in the morning. That is a really nice feature that Wild Courses allows you to kind of self-construct on there. So feel free to do that. Maybe it'll be a great way to communicate with each other. With respect to your topics, I know that this is a hard process, but I want you to start to narrow down your topics. And as soon as you want to get those to me, that's great. Some of you have already given me a rough idea of what your group is going to pursue, and there are some really good ones out there. I'm guessing the probiotics are probably going to pursue probiotics, but we'll see. Maybe not. Okay, cool. So I think that's most everything except for lab. We do have lab 1 tomorrow, so please get in and do the pre-lab prior to lab 1. And then come lab 2, we're actually going to flip everything on you just for fun, and you're going to have everything done before you get to lab on lab 2. And we'll talk a little bit about that tomorrow in lab. So I won't go too much into depth on that, but make sure you get your pre-lab 1 done for tomorrow. Sound good? Everybody feel solid on this? Now I have been told one thing. Just now, Haley told me she can't see the vodcasts on WIO courses. Have you guys been having that problem, everyone? Yeah, so I think what must be going on is that somehow I set a date wrong, so it's like not visible to you or something. I will check that out, I promise, and Haley's going to remind me. So thank you for bringing that up. All right, great. That is everything with regards to the boring announcements. We get to jump in, and we're going to begin with a question that previous students have told me, Rachel, this is kind of a no-brainer because what class are we in? Which one of the following killed the most Americans? Was it World War I, World War II, the Korean War, and the Vietnam War combined, or was it the flu between 1918 and 1919? It's your question. No, it's for real legit. The answer is just the flu. And what I bet you didn't know is that the influenza between 1918 and 1919 actually killed more than A, B, and C combined. Yeah. And so what war were we in during those years? World War I, right? And guess what? 116,000, roughly, a little over 116,000 Americans were killed in World War I. But guess how many Americans were killed by the 1918-1919 influenza? 500,000. And which one do we study more, right? It is crazy to think about the destruction that the influenza, the path that left in its wake. Absolutely disastrous. And so, yeah, we don't really study that much about it. It's interesting to note that that particular influenza was an H1N1, which is really interesting because what in our recent history did we see? Where did we see that again? Yeah, exactly. So some of you may have even suffered through an H1N1 virus, which actually refers to the characteristic spikes that are on the surface of the virus. So that being said, I want to briefly mention that one specific facet of microbiology is medical microbiology, and it is the study of pathogenic microorganisms. That is, these are microorganisms that cause disease. We say that they are virulent microorganisms. So virulence is the ability of a bacterium or virus or other microproteins to cause disease. Pathogenic microorganisms cause disease. So you guys will later take a course potentially called Pathogenic Microbiology, where you will focus in on these disease-causing microbes. Today we will just briefly touch on them. And I want to take you with me back to the turn of the century. At the beginning of the 20th century, if you access and look at the CDCs, you will find out that of the 4 leading causes of death, 3 of them were infectious disease. Number 1 was tuberculosis. You've seen the movie, right? Doc Holliday, Nicole Kidman in Moulin Rouge, right? Dying from tuberculosis, or also known as consumption. Pneumonia was a leading cause of death, as was diarrheal disease. And one of the leading causes of diarrheal disease was cholera. So this is actually a memorial in a little town called Selby that I biked through in England. And this was an entire town that was basically decimated by a cholera epidemic. And you can see that this was in memorial to that epidemic. And what they note on this plaque is that this was because they didn't have clean water. So what was happening to the water was a mixture of fecal contaminants in the drinking water. And so anytime you have that, you're getting the transmission of disease via what we call the fecal-oral route. And so diarrheal diseases are commonly transmitted that way. Now, this is not entirely a thing of the past, but certainly it was something that at the turn of the century was a leading cause of death. Tuberculosis, as I mentioned, also was. On this same bike trip, I saw this particular exhibit in the town of York, and it's actually showing the systemic case of tuberculosis where you could even see evidence of it in the spine. So throughout history, infectious disease has been a big thing, and between 1346 and 1350, 25 million were killed by the Bubonic Plague. This was a huge epidemic. And if you look even in more recent history, we can think about the fact that with 600 soldiers, Cortez conquered the Aztecs. What was his lethal weapon? Smallpox. Now, there's some debate about whether that was the only lethal weapon against the Aztecs, but certainly it was something that they were not familiar to seeing, so they were very sensitive to that particular Western disease. So we see throughout history a huge evidence, if you will, of infectious disease, but that doesn't mean that now, at this time, we don't deal with it, and we're seeing a rise of certain infectious diseases. If you look at the CDCs now, you'll see that 3 of the 4 are not infectious diseases anymore. So we sometimes consider some diseases to be eradicated, a word that I kind of see as a cuss word, but I'll talk about that later. And so now we kind of think of these new diseases as being trendy things. Maybe you've heard a little bit about valley fever. That was something that last year in California was a new disease that was a fungal pathogen that was causing death in a lot of people. You remember that? Yeah. Crazy, interesting spreads of diseases that maybe haven't been seen before. If you guys do choose to pick up the book Fever, you're going to read a little bit about why that might be. It's going to be really interesting. Some of you may have already done so. So what are some other things that we have to think about now? And I thought I would pick a little bit on, I think it's Abby, right? You go by Abby. Are you here? But it's Maria, but you go by Abby. Is that right? No. Okay, I'll talk to her on here. But she says she loves to travel. I guess William, you also like to travel, right? So what's like one of the issues with international travel and disease spread that we didn't have in 1906? Yeah, a lot easier for disease to spread across international borders. A good example of that is in 2003, I actually traveled to China, and I spent time in Beijing and Xi'an. And when I left, I ate breakfast in Beijing, I ate lunch in Tokyo, I ate dinner in San Francisco, and I ate a late-night snack in Laramie, right? And yeah, I know, right? And you think it's totally true. And if you think about it, 2003, what was going on right then? SARS. Yeah, I could have come back with SARS, and then I would have spread that disease throughout every layover that I had. So it's really interesting to think about how international travel plays into disease transmission. We also talk about other things, and I think one of our groups is going to talk about vaccination as their poster topic. But also many old diseases are making a resurgence, and particularly those vector-borne diseases. What is an example of a vector-borne disease? Something that maybe is, Esteban? You looked like you had something. Sure, good. Malaria, or one that's kind of been a thing in our area recently. West Nile, right? I know. So interesting stuff to think about. Lyme disease making a resurgence. Everybody remember when President Bush got Lyme disease, that was something that we started talking about, the increase in vector-borne diseases. So that is definitely yellow fever, things like that. Also, we're finding out that many chronic diseases, such as peptic ulcers, are being shown to be caused by microorganisms. We didn't know before that there was a link with the microbial world. Now, there's more nuances to this than even we thought in 1983, when people finally realized that Helicobacter pylori was certainly strongly associated with peptic ulcers. Prior to that, what did we think gave you an ulcer? Stress, right? And they contribute to it, but we now understand that there is at least a link with the microbial population, even though that's more nuanced. In fact, we now understand that Helicobacter pylori in the right concentrations in your gut will actually prevent acid reflux, but in the wrong concentrations will cause peptic ulcers. So we're understanding the nuances better there, but we're seeing those links. Also, hey, you guys even remember 2006, the summer of 2006? We figured out that what was causing most cases of cervical cancer. HPV, and we started to see the vaccination for HPV. So we're starting to figure out those links a little bit better, a little bit more efficiently. And as all of you know, antibiotic resistance is huge. And you might have seen, I've been tweeting out a few things regarding a conference right now that's going on in Washington, D.C., devoted largely to antibiotic resistance, because this is probably, in my mind, the biggest thing that we face right now in the microbial world, right? Selective pressure. Okay, that was a lot in a very short breath. But I want to introduce you, right, to pathogenic microbiology. But I also want you to know that by no means are most microorganisms pathogenic. In fact, these are like the Hollywood of all microbial life. They get all the attention, you know, but by far they are not the entire population, right? They really don't represent the entire population. So in fact, most microorganisms are not pathogenic. And as we know, our microbiome protects us. So those microorganisms that are health-giving, that live on us and in us, are very protective to us. And we also know that microorganisms are great study or model organisms. That is, in a lab, we can study E. coli, and we can make sort of trending conclusions about what might describe other forms of life. And there was a very famous scientist, his name was Jacques Monod, and what he said, his quote is beautiful. He said, "What is true of E. coli is also likely to be true of an elephant." So that we can study something quick to reproduce like E. coli and then make generalizations about, say, an elephant. You know, there's some nuances to that, but it's pretty true, right? We can still study glycolysis in E. coli and know something about glycolysis in us. That being said, we are about to switch to our game for the day. Who has questions about pathogenic microbiology and or any of our other introductory material? I know I got a couple emails saying, "Help, Rachel." Marissa emailed me. She's like, "I can't answer the last 4 questions of the homework right now. I totally don't know where I'm even supposed to find that stuff." So right now is when we are going to answer those 4 questions. So what I want you to do is I want you to find someone who was organized enough that they printed the amino acid table. So if you printed that amino acid table, you know, find that, hold it up. Cool. Share it with everyone. Okay. Wonderful. Now, that is all you're going to need, notes-wise. For this game, I am going to be posting the complete notes, so you don't need to, like, be scribbling a lot right now. But I want to say, like, there's a little caveat to this game. Some of you are going to be like, "Whoa, this is totes review for me. Like, I don't even need to listen right now. I know this stuff." So you guys need to be the teachers in your group, right? Some of you, like Erin over here, has already taken biochemistry, so she is a huge resource for you. Anybody else already taken biochemistry? Okay, see, so you guys are definitely resources. Remember that there are people in your group and in the class who have that kind of background. How many of you, the highest chemistry you've ever taken is Gen Chem, right? So you guys don't have as much chemistry, so you're going to want to talk to the people who have a little bit more. Some people are through OChem. So I want you to know that not everyone is on a level playing field today. So if someone's shouting out answers like, you know, they're the shiz-niz, please know that not everyone is at the same level. And I look to you to kind of teach each other within your groups. We're going to be talking now about the macromolecules. So as we get started with this game, again, don't feel you have to scribble down notes. I will be posting this. What does it mean to be macro? Big, okay? So big molecules, macromolecules. That is what we're going to be focusing on, and there are 4 groups of these very large molecules. There are the proteins, the carbs, the nucleic acids. Now, what do we usually say nucleic acids? What do we usually call those? DNA and RNA, okay? So that's the nucleic acids. And then finally, the lipids. These are our 4 classes of macromolecules. Now, does that mean that there are no small molecules in a cell? No, right? For example, we know, some of us are all too familiar with lactic acid, a small molecule within a cell. Pyruvate, the precursor to lactic acid, we're familiar with that as a small 3-carbon molecule. Heck, Water is all over in the cell, a small molecule. So macromolecules, although they're famous in a cell, are not the only molecules that are in a cell, but they're going to be our focus right now. So what I want you to do is with your group now, and you can be as noisy and rowdy as you want, I want you to list at least 3 protein functions. And when you're done with your list, I want you to shout out, I don't care if you stand up or whatever, let me know you've got your 3. Okay, go. Always running late. That's right.
Our transport phenomenon teachers did a long weekend.
Uh-huh. Yeah, I manage you guys are getting worked over. No. Yeah, you guys are not turning this in. You can put it on the back of your hand if you want, you know. You've got it? All right. All right, this group is, okay, so Natasha's hand was the first up. I think we've got 3 functions. Let's start with 1 right here with your group. What's the first one you have? Transporters. Okay, proteins can be transporters within a cell. That is to say that they span the lipid bilayer, the membrane, right? They allow things to go in or things to go out. Transporters. Fantastic. Who's got one other than transporters? You guys, go ahead. Energy. Okay, I like that. As far as serving as a source of energy for the cell, can proteins be a source of energy? Yes. Okay. Now, how does a cell feel about them as a source of energy? Right? You know, it's only macromolecule that actually has to be, it's only energy source, let's say that actually has to be detoxified prior to use. So that's, you know, what urea is, right? Urine, is that we have to detoxify proteins before we can incorporate them as energy sources. So yeah, definitely a good answer, but we'll say that the cell doesn't prefer it, and it can be a thing. Okay, cool. What else? What you got, Ryan? Okay, enzymes. Enzymes are catalysts, right? An enzyme is one particular kind of protein that acts to speed up reaction rate. So one kind of protein are enzymes, my favorite kind actually, right? Rockin'. They're speeding up reactions. Okay, who's got something else? Yeah, what you got? Receptors. Marvelous. So next to those transporters that this group came up, we might see another membrane-spanning protein that is a receptor. Maybe it binds to what we call a ligand, which sometimes we think is the voices of the cells, and that ligand might bind to a receptor. It triggers conformational changes in the receptor, and then it triggers intracellular changes. Excellent job. Okay, great. Anything else? Okay, they're comprised of amino acids. So proteins are made up of how many amino acids? 20 standard or common amino acids. Good. Yeah, go ahead. Wait, was that yours? Yeah, no, you're fine. I saw somebody else's hand. Okay, what else? Yeah. Structure. Excellent. So this is something that maybe we tend to think about within eukaryotes, structure, but that's also true in prokaryotes. And we're going to see that, in fact, actin filaments are going to play a role not only in structure but also in motion. And there are other proteins, right, that are responsible for structural conformation, right, within the cell. Absolutely true. Good, good. Anything that we missed? What about their role in fighting infection? Yeah, proteins are also, or what I should say is, antibodies are a type of protein. Yeah, so they also play a role in our immune function, right? They're a specific kind of protein. They're called a glycoprotein, which means that they have both carb and protein. But antibodies are absolutely proteins. Isn't that cool? And then what about the fact that proteins can regulate gene expression? Yeah, isn't that cool? There are literally proteins that can bind upstream of genes and turn them on or turn them off. Pretty cool. So proteins have so many functions within the cell. You guys listed a lot of them. And again, I will be getting this out to you. You can add to this list when I get this sent to you or posted on Wild Courses. But we hit on nearly everything on that list. I'm not sure we got to the point of mentioning that certain hormones are actually peptide hormones. So that's actually one that we could add to that list. But for the most part, you did an excellent job. And we came up with this idea that proteins are made up of amino acids, right? So the little subunits that repeat to make up a protein are amino acids. There are 20 standard ones. There are some other unusual ones, but there are 20 standard ones that make up a protein. And so I actually have a model with me today where I can show you the structure of an amino acid. Essentially, you've got a central carbon, and that's up there too, right? A central carbon. That central carbon is attached to a hydrogen, right here a hydrogen. It's also attached to an R group. What does that mean, an R? Okay, good. Yeah, exactly. It may be a carbon chain. For this purposes of our characterization of amino acids, it means that it is some functional group, right? Some functional group that's attached at R, okay? Because sometimes it's a carbon chain, but sometimes it's a simple hydrogen that comprises that R group. And then what do we call the group on the right, or this guy here? Carboxyl. So there's actually a carboxylic acid group. And then on the other side here, or on the left up on the overhead, that is an amino group. Excellent, good. Now here's the thing. Every single amino acid has a personality. It gets its personality not from the upper part of the molecule, because the upper part of the molecule is completely the same in every single amino acid. So right here, this is retained no matter what amino acid you're looking at. They all have an amino, a carboxyl, and a hydrogen. But the place that they differ is in that R group. And it's that R group that gives them their personality. So what I want to talk now about is their personality. And I want you guys to figure out, as a group, what amino acids have what personalities. So in a minute, I'm going to put you into separate groups. But what I want to know is what amino acids are, first, hydrophobic, hydrophilic, basic, or acidic. And here's how we're going to break up. You guys on this side of the room, all of you in this side, are going to look at those that are hydrophobic. Now, real quick, what does that mean? Water hating, right? Water hating, or they don't like to be around water. You guys are going to work on hydrophilic, water loving, okay? Now, you guys up front here, I want you all, I'm giving you the hard one, basic, okay? And then you guys get another hard one, acidic. And I want you to look now at your amino acid tables. If you don't have one, be real rude and shove your neighbor and ask to look at theirs. And I want, as a group, I want you to talk about what does it mean to be acidic? What does it mean to be hydrophilic, right? What does it mean to be hydrophobic, okay? So how can you approach this hydrophobic thing? Like what do we know doesn't like water? Lipids. Yeah, good. Lipids, so fats, anything that's fat. Right, which also are, lipids and fats are. Yeah, good. So find your R groups that look like lipids, right, that look like fat, or that are lots of hydrocarbons. So if you see lots of hydrocarbons on those R groups, good. Are you guys figuring out, how do you know if something hates water? Like what kinds of things hate water?
Cats.
Yes, exactly. Right? Except for one of my cats, man. She loves it. She sticks her head under the faucet. It's ridiculous. But what are some substances, right? Non-polar, good. Lipids? Good. How might you recognize a lipid just in its chemical composition? What are they made out of? Yes. Carbon chains bound to? Not so much oxygen. Not so much nitrogens. Hydrogens. So think of this. Fats and fuels in general are hydrocarbons, right? So look for those R groups with that personality. Okay, you guys are on the hard one, right? Acids. How do you know, like, what does an acid do in solution? Yes, it gives up not so much a hydrogen, but a proton. Good. So it gives up a proton, meaning that once it's given up that proton, what charge does it have? Right? Now, what do bases do in solution? What do they do in solution? Yeah. So if something takes on a proton, excellent. Okay. And I see Aaron's teaching. Okay, great. All right, I think we have some good answers ready to go. So hydrophobic, you guys over here, we're coming up with that. Which amino acids, now I'm going to pull up the table, and you can tell me which of these amino acids are major water haters. Of course, I got the answer, cats, water haters. But besides that, which amino acids are, hey, which amino acids could we call the cat amino acids? Yes, I love that. What are they? Okay, maybe lysine, because I know what your rationale is. Tell me your rationale. Carbon chains. They're looking for hydrocarbon chains, right? Hydrocarbons, right? Hydrocarbons, fuels, or fats, or oils hate water, right? So they're looking for the R groups that are hydrocarbon chains. And they said maybe lysine. And I like that answer because look at all those hydrocarbon chains, but what busts the deal? The amino group. Look at that nice positive, like, hey, I've got a positive charge. Can I associate with your partial negative water? Right? Yeah, it likes to associate with that partial negative. So if we took off the amino group, we could say that lysine was hydrophobic. But with its amino group on, what is it? Hydrophilic, exactly. You guys can celebrate over there and find lysine as a highly hydrophilic, actually, because of that amino group. But that being said, which one of these amino acids don't have that water-loving group and just have hydrocarbons? Leucine, right? How about this guy? Leucine. How about isoleucine? What else? Valine. Valine is another one there. It's got the V-shaped R group. That R group, very water-hating. Excellent job. We could probably find some more in that list as well. How about this one? Oh, yeah, because what's that? Yeah, like a benzene ring, and we can see that all is hydrocarbons as well, right? So that one's also phenylalanine, very, very water-hating, okay? So now that being said, you guys already told us that lysine is one of our water lovers, but what else is going to be majorly water-loving? What else is going to love to hang out? Besides that positively charged lysine, what else?
Anything with a charged group on it.
You bet. Anything that has a charged group is going to love water. Because remember that water is a molecule that has an overall what? It has partial charges, good, Bridger, but overall it has, what's that sexy term? Dipole moment, right, good. So remember that it has bent geometry with a partial negative charge on the oxygen and partial positive charges on the hydrogen, thus giving this an overall dipole and making water love to interact with any R group that is charged. An R group like lysine, if it's R with a positive charge, like lysine, if that's the R group, it's going to love associated with a partial negative charge of oxygen. If you have something like, for example, an R group with a negative charge, what's a good example of that? Okay, good. Glutamate or aspartate, also known, in a minute you guys are going to tell us, but glutamate and aspartate both are going to love to associate with that partial positive charge on water, okay? Now, if you guys leave with nothing else today, you're just zoning out, major, that's all right. I want you to leave with this idea. Like dissolves like, okay? Because this idea is so important. If we have something that's polar, something that's charged, it's going to dissolve in water. If we have something nonpolar, a hydrocarbon, no go. It doesn't like, it's a cat amino acid, right? Absolutely. So now we're left with what are bases and what are acids. And you guys had phenomenally good answers, the best I've ever heard. So you were rocking my world. Why do we know something is an acid? It gives up a proton to solution, and thus it has what charge? Negative. Excellent. So what are our acidic amino acids? Two of them. Excellent. Also known as aspartic acid and glutamic acid. Right? So those are our two acidic amino acids are aspartate and glutamate. And let me see if I can get my pen up here to show you those. Right here. See that negative charge right there? So the R group on aspartate and the R group on glutamate both are negatively charged, therefore making them the acidic amino acids. Therefore, what are our basic amino acids? Excellent. So lysine, basic, water lover, and we have arginine. Actually, guess what, you guys? Arginine is the most base amino acid there is, and I love it. It's the pirate amino acid, right? Arg. Yeah, arg. Okay. It's the most base. Right? Got to give them personalities because that's how we get to know them. Now, histidine is actually my favorite amino acid because it's a proton slut. It likes to give up and take hydrogen. It will actually take on a proton, give up a proton, take on a proton, because for those of you who speak chemistry, if you don't worry about it, its pKa is very near neutral. So it's always given and taken and given and taken protons, right? That's what it does. And therefore, it is super jazzed at being at the active site, Brent, of enzymes. Yeah. If you don't see a histidine at the active site of an enzyme, it's a really odd enzyme. Histidine really gets around. It's in a lot of different active sites, okay? It's a really cool amino acid. Okay, cool. So this allows us to start to understand the personalities of amino acids. How do we feel? Do we feel jazzed on this? Okay, because if you do, you guys are rocking. This is the hardest chemistry we do in here. So if you're feeling good about this chemistry, then you're feeling good about the hardest chemistry that we do. Okay, good, good. Little more in the way of questions for you, but first, I want to mention that I will not ask you to memorize the amino acids. So let me say that again. I will not ask you to memorize the amino acids. On the exam 1, you will find that table that you are flying around in your hands right now. That exact table will be stapled to the back of your exam. So I'm not going to ask you to know, to memorize their structure. But what I am going to ask you to know is their one-letter abbreviations, because we talk in one-letter abbreviations all the time because we're lazy. And so all the time when we write a sequence of amino acids in order as they're linked to one another, we use their one-letter abbreviation. And so we will write something, for example, see how fast you guys can think with me. We will write something that looks like this: V, F, F, I, M, dot, 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 and so on forth. And that sequence of amino acids is what? Very hydrophobic, hydrophilic, basic, acidic. Very hydrophobic. Excellent. Because that is valine, isoleucine, leucine, phenylalanine. And we know that those are big-time water haters. Big old hydrocarbon R groups on them, right? So that's why I'm asking you to know their one-letter abbreviation. So here's what I want you to do. When I post this later today, I want you guys to get the game answers. I want you to print this off, maybe even in color, splurge a little. And I want you to put it up somewhere that you look all the time, okay? So, you know, over the top of your bed, in front of the sink where you do dishes, you know, in front of your toilet, I don't know, somewhere that you spend a lot of time. And I want you to start to learn the one-letter abbreviations, okay? So you don't need to memorize the structures, but you do need to know those one-letter abbreviations. That being said, let's talk more about some of these amino acids and what they can do. So, if you have two amino acids, they can link to one another. And they can do so by forming a bond between the amino and the carboxyl of two neighboring groups. So if we have, let's see if I put this together in the right way. If we have the carboxyl of one amino acid, it can bind to the amino of the neighboring one. That's also shown up here. Notice that the carboxyl is in fact binding with the amino group, and we're forming a bond. And what I want you to discuss with your group right now is what is that bond called, and what molecule is lost when that bond is formed, okay? This one should be a quick one, so do it. I'm working to make the bond here. Anybody think they know? Already? You guys got it? Okay, hit us. Oh, okay, so the name is a peptide bond. Anybody know the nerdy chem? Okay, there's so many chem engineers in here. What is the nerdy chem name? It's an amide bond. Okay, so peptide, though, is what we always call it. We call it a peptide bond in the biology world, but it's also an amide bond. Okay, and then the question is, what molecule is lost? Water, okay? So I've got my volunteer over here. I've just made a peptide bond, and why can't I remember your name? Alex is totally volunteered over here. He just was telling me I'd like to volunteer, and I've got his water molecule. Looking good? That's what we cleaved out of there. Sweet. All right. Thank you for that. So to story time. Don't give it away if you know the story. Years ago, I did that demo, and I picked on a football player. He was also a wrestler. He did both, and big guy, right? And so I hand him what's left, and I say, you know, what do you have there? And he says, little balls. And I said, that's something you don't want to divulge. And by the way, we became best friends that day. He became a teacher. So, just so you know. It's a thing. So you guys are absolutely right. Water, and the name of the bond is a peptide bond. Okay, I've got another one for you, I promise. So now we have this. What do we have here? Now that I've lost my water, Alex is holding on to that for me. What do we have now? We have a peptide bond. How many amino acids in it? 2. What might be a word we would use for 2? Di. So this is a dipeptide, right? What if I added a third one via a peptide bond? Tripeptide, and so on forth, until finally Zach tells us we get a polypeptide, right? Between 2 and 20 amino acids is typically called an oligopeptide. Then above 20, we call that a polypeptide. Okay, so we eventually get a very long string of those. And I'm sorry, I don't have enough of these to make a full polypeptide, so use your imagination. There isn't much you can't do with styrofoam toys and pipe cleaners. And so now we have a polypeptide. Imagine that each one of these, right, is an amino acid linked together via a peptide bond. So maybe this is valine, isoleucine, phenylalanine, right? All of our hydrophobic amino acids right here. And then maybe we start getting lysine and arginine and lysine-arg and lysine-arg. And what is that region? Hydrophilic, but also basic. Right, excellent job. So we've got characteristics of the entire polypeptide chain. So the characteristics of the individual amino acids, their personalities. If we have a cat region, right, then we know it's going to cower from water. It's going to want to hide on the inside of the protein as opposed to exposed to the exterior aqueous solution, right? So we know that the personality of individual amino acids is going to dictate the structure that this polypeptide takes on. So eventually we're going to see that folding into a responsive structure to that. The structure that I have here is called the primary structure. Primary literally refers to the amino acid sequence. So when I give a sequence like valine, phenylalanine, isoleucine, methionine, that is a primary structure. Secondary structure is when this starts to fold. So maybe this region wraps up and it forms a helix, right? It forms this helical structure, maybe, kind of, there we go. It forms an alpha helix, okay? So a secondary structure is the local repeating structure of neighboring amino acids. But now maybe we get a region that's helical there, maybe we get a region here that forms a beta sheet. That is a more extended region like this that kind of goes back and forward on itself and hydrogen bonds to hold itself together. Those are both secondary structures. But now we also know that this helix is going to orient next to the sheet in three-dimensional space. And what's that called? Tertiary structure. But it doesn't stop there. It keeps getting good because many proteins don't have just one polypeptide chain, but instead they have two, or they have three, or they have four, and maybe this is our other polypeptide chain that itself has taken on unique secondary structures. Maybe this one is primarily alpha helix, so it forms a lot of helices, or it forms one, well, kind of one long helix, and how that associates in 3D space next to another polypeptide chain is called quaternary structure, okay? So two polypeptide chains associating with one another is quaternary structure. What really famous protein has quaternary structure? Hemoglobin. It actually has 4 polypeptide chains that all associate with one another. So we have these 3 levels of structure: primary structure, 4 levels, secondary structure, tertiary structure, and finally quaternary structure that describes these. Now, we tend to draw this with something that we term a ribbon diagram. And this ribbon diagram looks kind of like this, where helices are drawn like this, and then sheets are drawn as arrows, where you see a directionality in the sheet, showing you that in this case, the sheet is an anti-parallel sheet where one strand goes one way and the other strand goes the other way. And that overall is showing a secondary structure. Now, what can't I show you very well on the board like that? Tertiary structure, right? 3D in space kind of structure, which is why just as soon as you guys leave here, I imagine you're going to go dork out to something called the Protein Data Bank, or if you've got your phone with you right now, look at Protein Data Bank. You can look up any protein in there, and you can look at it in 3D space. You can turn it around and roll it around and look from one side and the other side, and you can see the 3-dimensional structure as well as the secondary structure and get information on the primary structure. In fact, you can download it onto your computer and click on it and see which amino acids come one right after another to make that primary structure. So I highly recommend dorking out with Protein Data Bank when you have a chance. But real quick before you leave today, I know you're getting ready to go here. Could you guys quickly tell me, what is it? So primary structure dictates secondary structure, right? We know that the personality of the amino acids dictates the secondary structure. Secondary structure dictates tertiary structure. And of course, those lower levels of structure dictate quaternary structure. But I want to know what dictates primary structure, the sequence of amino acids. Yeah, good. RNA. And what dictates the sequence of RNA? DNA. So leave today with the central dogma of gene transfer in your mind, which is DNA dictates RNA, which dictates protein. Okay, have a fantastic day, you guys. We're going to do more game action next time. We're going to be looking at carbohydrates next time. So, as you're leaving, what is the kinky amino acid from that table? I think that might be on your homework. Proline, right? It's the only one that binds to the backbone again. It kinks. It's my second favorite.